Hello, we are Group 10, and the topic that we are bringing to you today is on phloem translocation. Let's move on to the theoretical aspects. Glucose is made in the leaves by photosynthesis. Glucose is converted into sucrose in the leaves, which then enters the phloem vessels as do amino acids. They then need to be transported around the plant to every single cell. The areas of the plant where sucrose is made are called the sources, and where they are delivered are called the sinks. The phloem uses active transport because the sucrose moves against its concentration gradient from the lower concentration where it is made and to higher concentration in the phloem cells. The phloem transports manufactured food like sucrose and amino acids from the green parts of the plant like leaves to other parts of the plant. The process is called translocation. Phloem consists of sieve tube, companion cell, fiber, and parenchyma. A sieve tube consists of a single row of elongated, thin-walled cells. Just like the name suggests, it has a wall perforated by minute pore-like sieve. A mature sieve cell has a thin layer of cytoplasm which conducts manufactured food from one cell to another. The sieve tube cell has a degenerated protoplasm, which means that it needs another to carry out its vital processes. This is why a sieve tube element has a companion cell. The companion cell has everything that the sieve tube doesn't, such as abundant cytoplasm and nucleus. It has an extremely thin but flexible cell wall, so there's no hassle in keeping the sieve tube cell alive. The fiber cells help to give structural support, and the parenchyma store the food and other substances. The fiber cells are the only type of cells that are dead. The mechanism of phloem translocation is pressure flow hypothesis. This idea was put forward by scientist Ernst Mach in 1930. This idea is based on the movement of fluid. Other parts of the plant that were involved in phloem translocation except phloem are the xylem, the source cell, and as well as the sink cell. Let's take a hypothetical situation where we are considering a growing fruit cell as the sink cell and a mature leaf as the source cell. The evaporation of water in the leaf from the process of transpiration creates a constant tension at the leaf. This tension results in pulling up of water from the roots via xylem. Next, the source cell loads sucrose into the phloem sieve tube, which results in reduction of water potential in the sieve tube. Then, the water comes into the sieve tube from the xylem by osmosis. This results in increasement of water potential in the sieve tube again. The uptake of water generates a positive pressure that forces the sap to flow along the tube to the sink cells. The pressure is relieved by the unloading of sucrose and the consequent loss of water at the sink cells. Now let's move on to the experimental procedure on phloem translocation. The Girdling experiment was what we followed. The main objective of our experiment was to find out whether the xylem or phloem tissue is involved in translocation. We followed the hypothesis that the phloem is responsible for the translocation of food because the phloem is present outside of the xylem. So, when a ring of bark is removed from a woody plant, the woody xylem part remains intact, which causes the water and the nutrient to reach the leaves. The method that we followed involves the removal of the tissue outside to the vascular cambium, which involves the bark, cortex, and the phloem in woody stems except the xylem. Xylem is the only remaining tissue in the girdled area which connects upper and the lower parts of the plant. This setup is placed in a beaker of water. In below diagram, you will be able to see the first experimental setup that we did in this experiment. We carried out 4 experimental setups in order to get the successful observation. Now let's move on to our first experimental setup. We got 2 branches of same length and same width and in one branch we removed the area that was mentioned in the experiment, and then we put it in a basket, in a clear basket containing water, and we kept it for a while. But we were unable to observe the required observation as it resulted in the death of 2 branches. So we moved on to our second experimental setup. In our second experimental setup, we chose a big mango tree, and then we carried out the same procedure in which we are removing the bark which is consisting of the phloem while keeping the xylem safe. This also did not give any results after observing it for several weeks. So we moved into our third experimental setup, which finally gave us the successful observation. In this setup, our member Takshila chose a medium-sized anoda tree. In this tree, she carefully removed the bark area which consisting of the phloem. Now you can observe how she removed the bark area of this branch. She has also kept a rigiform board behind this setup, so you can observe it carefully. After around approximate 3 weeks of observation, we were able to get the successful result that we desired. In this picture, you can see the above area has been swollen while the below area is in its original size. Now let's see the conclusion that we reached after this observation. We reached into the conclusion that phloem is responsible for the translocation of food because the phloem is present outside the xylem. After girdling, photosynthetes get accumulated just above the girdle region and are not transported to the other parts below the girdle. This experiment shows that phloem is responsible for the food transport and it doesn't affect water transport done by the xylem present in the inner side of the vascular plant.